In the universe, there are many important things that create the fundamentals of how things are today. Amongst one of them is the creation of life. But how is life actually created? There are many ways that life can come into being. Different organisms use different methods to reproduce and continue their lineage or species. Some of these include fragmentation, where they split themselves, and each of the segments create new organisms with identical DNA and properties. Budding is when organisms grow many parts of themselves that eventually split off and become separate and individual organisms. These are amongst the few ways that organisms can begin life. However, these are all only asexual reproduction. Humans, as well as many other living things, do not use asexual reproduction to reproduce. They use sexual reproduction. This is when the gametes of two parents come together and fertilize, beginning a process called meiosis, the main topic of this video.
Meiosis is the creation of 4 haploid daughter cells from the fertilization of 2 gametes. Meiosis has 2 cell division stages, each containing 4 phases. The first stage of meiosis contains the phases prophase 1, metaphase 1, anaphase 1, and telophase 1. The second stage of meiosis quite literally has the same phases. The only difference is the Roman numeral that follows the name. The 4 phases of the second stage of meiosis are prophase 2, metaphase 2, anaphase 2, and telophase 2. Gametes are defined as sex cells. In humans, there are 2 kinds of gametes: eggs from females and sperm from males. Eggs, the female reproductive cells, are also called ovum, or in plural terms, ova. They are contained in the ovaries of a female. Sperm, the male reproductive cells, are also called spermatozoa. They are contained in the testes of a male. First, the sperm has to go through a series of obstacles as it enters the ovaries. It partakes on a journey and is motivated on completing one mission: to get to the egg. Once the sperm completes the race against other sperm to get to the egg, the process of meiosis can begin. Fertilization occurs when the haploid sperm cell unites with the haploid egg cell, forming a diploid zygote. The zygote is what forms a new individual. The term haploid means that the cell has 23 chromosomes, and the term diploid means that the cell has 46 chromosomes. Egg and sperm cells both have 23 chromosomes, and the zygote they form has 46. In prophase 1, the chromosomes condense. The chromosomes that were replicated during interphase consist of 2 sister chromatids that are joined together by a centromere. A sister chromatid intertwines with another sister chromatid. The pair of sister chromatids is referred to as chromosomes. The chromosomes join together, creating homologous pairs in a process called synapsis. The strands from each parent cell also go through a process called crossing over, which is a term used to describe the exchanging of genetic information between homologous chromosomes during synapsis. The nuclear membrane dissolves during prophase 1, and the centrioles move to opposite sides of the cell. Spindle fibers also form during prophase 1. The nuclear membrane dissolves during prophase 1, and the centrioles move to the opposite sides of the cell. Spindle fibers also form during prophase 1. In metaphase 1, the homologous pairs line up in the middle of the cell. The spindle fibers attach to the homologous chromosomes. In anaphase 1, the homologous chromosomes are split apart by their centromere and dragged to the opposite poles of the cell by the spindle fibers. In telophase 1, cytokinesis, which is a division of the cell, happens. The result is 2 haploid daughter cells. That is the end of meiosis stage 1. After meiosis 1 has been completed, the 2 daughter cells would have been formed. Meiosis 2 will then begin with both cells.
The phases in meiosis 2 are similar to the phases in meiosis 1, just less eventful, but equally important. The stages also show close resemblance to the process of mitosis. Meiosis 2 starts off similarly to meiosis 1, except instead of prophase 1, it starts with prophase 2, and instead of starting with 2 haploid cells, it starts with 2 daughter cells. In prophase 2, the nuclear membrane dissolves, and the centrosomes move to the opposite poles perpendicular to before, and new spindle fibers forms. Unlike meiosis 1, no crossing over happens in this stage. In metaphase 2, The spindle fibers from opposing centromeres attach to the chromosomes at the centromere and align them along the cell equator. This phase results in the chromosomes being aligned in the center of both cells. In anaphase 2, the spindle fibers contract and separate the sister chromatids. These chromatids, now called chromosomes, move to opposite poles. In telophase 2, the chromosomes decondense and become less visible. A new nuclear membrane begins to reform as each cell divides, resulting in 4 daughter cells. You may be familiar with this process. It's called cytokinesis. From the beginning to the end of the entire process of meiosis 1 and meiosis 2, the end result will yield 4 haploid daughter cells that will then perform mitosis over and over again.